All right, so in this section, we're going to specifically break down the process of mitosis. Mitosis is going to be responsible for creating new cells in the developing embryo, fetus, and child. It's also going to be responsible for the replacement of cells in adults. During mitosis, the cell that divides is called a parent cell, whereas the two cells that are going to be produced from the end are called daughter cells. This process is referred to as duplication division, since the two daughter cells are genetically identical to the parent cell. This is going to be pretty important. Does anybody know why we would want our daughter cells to be genetically identical to the original parent cell? So take a moment, think through that. Maybe reach out and let me know what you think. So DNA is going to be replicated during the S phase of interphase. At the end of S phase, each chromosome is going to contain two identical parts called sister chromatids. So this is what goes into the S phase. This is what comes out of the S phase. We start off with a single chromosome. We end up with two sister chromatids attached at the centromere. Eventually, these two sister chromatids are going to divide, but during this replication process, when this single chromosome replicates, we have two completely genetically identical sister chromatids. When they divide, we will have two completely separate but genetically identical daughter chromosomes. As mitosis begins, the chromosomes are going to condense down. So we had that very loosey-goosey chromatin that was sort of all bunched around, filling up the nucleus. During the process of mitosis, those chromosomes are going to condense down. Following the separation during mitosis, each chromatid is going to be called a chromosome. Each daughter cell is going to get a complete set of chromosomes and will be considered diploid. So here's an example. We started off with a diploid cell. We have 1, 2, 3, 4 different chromosomes. You can see that these two, this short one here and this short one here, match together. This long one here and this long one here match together. We have two maternal chromosomes and two paternal chromosomes. So here we have, let's call these chromosomes, the short one can be 1, the long one can be 2. So we have two number 1 chromosomes and two number 2 chromosomes. We have one paternal, one maternal, chromosome 1, one maternal, one paternal, chromosome number 2. So each daughter is going to get a complete set of chromosomes. So what's going to happen is each one of those chromosomes is going to duplicate and produce a sister chromatid. So here we see that first paternal chromosome 1 duplicated, maternal chromosome 1 duplicated, maternal chromosome 2 duplicated, and paternal chromosome 2 duplicated. When each one of these duplicated, they formed sister chromatids. Now we are going to undergo mitosis. In the process of mitosis, each one of those chromosomes is going to separate. Each one of those sister chromatids is going to separate. What we end up with is one cell that has a paternal chromosome 2, maternal chromosome 2, paternal chromosome 1, maternal chromosome 1. We end up with another cell that has a paternal chromosome 2, maternal chromosome 2, paternal chromosome 1, maternal chromosome 1. In other words, are these daughter cells genetically unique or genetically identical to each other? So take a moment and compare them. Are they identical or not to each other? And then also compare them to the original parent. Are they identical or not? So there are different phases of mitosis that are going to carry out this reduction division. Mitosis is going to be divided into multiple phases: prophase, prometaphase, metaphase, anaphase, and telophases. Now these stages are continuous. These are man-made stages. The cell does not start and stop and start and stop at each one of these phases. The only time the cell stops in its cell cycle is during those checkpoints. Otherwise, this is one continuous phase with the cell seamlessly transitioning from one phase into the next. There's no noticeable interruption. So it's sometimes difficult to determine, hey, is this cell in metaphase? Is this cell in prophase? It's in anaphase. What stage is this cell in? Can be a little bit difficult to determine. because they are all continuous, these stages, one with another. We are going to start off with the prophase. Prophase is the first stage of mitosis. During this, our chromosomes are going to condense down. So the centromeres, the centrosomes have duplicated. We're going to see the centrosomes are going to be the microtubules that are going to be responsible for controlling and organizing this entire process. These microtubules are going to condense or have duplicated. They're going to then start to migrate to opposite ends of the cell. We'll have one stay here and one is going to migrate over here. Spindle fibers are going to start to appear. These spindle fibers are going to branch out from the centrosomes. These are going to be the microtubules that are going to help push, pull, and direct the chromosomes to where they need to go during this process. The nucleolus is going to also start to disappear, and the nuclear envelope is going to fragment. If you remember way on back when we talked about the different parts of the cell, we talked about how the nucleus is surrounded by a nuclear envelope. So this region right in here is our nuclear envelope. This is going to start to dissolve and disappear so we can get at those chromosomes. Those chromosomes are normally protected inside of that nuclear envelope. So what we're going to do when we're exposing our DNA to the rest of the contents of the cell, we're going to condense them down, bind them tight, pack them up nice and tight, and make sure that they are safe for the travel process. The nucleolus is going to disappear, the chromosomes are going to condense, and those chromosomes are going to each be composed of two sister chromatids that are held together at the centromere. During prometaphase, the spindle fibers are going to start to attach to those centrosomes, and they are going to start to push and pull those chromosomes to where they need to go. Chromosomes are going to be pretty randomly located in the nucleus, so we need these spindle fibers to attach them and address and push them wherever it is they need to go. And here is where they need to go. In metaphase, we're going to see those chromosomes are going to line up down the equator. Those chromosomes are going to line up one right after another right down the center of the cell. We're going to be getting ready to split each one of these chromosomes. So those chromosomes attached to the spindle fibers are going to line up at the metaphasial plate. They're going to line up right down the center along the equator. When this line forms, it's also known as the metaphase plate. This is going to be our metaphase of mitosis. This is where the M checkpoint is going to occur. Before we can divide, we need to run down the line and make sure that all of these spindle fibers are firm and secure. And if we have run through our seatbelt check, we make sure that these spindle fibers are attached firmly in place, then we can proceed to the next stage, which is anaphase. Anaphase is when the centromeres are going to divide. What's going to happen is that those centromeres, those points where those sister chromatids were held together, is going to dissolve. Those spindle fibers are going to retract. They're going to pull those sister chromatids apart and separate them, one going towards each of the centrosomes that are on opposite sides of the cell. As they pull them, they're going to separate those chromosomes. So we're going to separate those sister chromatids, and we're going to have, right down either side, daughter chromosomes. Each one of these is separate and now by itself. Once we have separated those daughter chromosomes, we're going to begin the process of telophase. Telophase is going to be the start of forming a new nuclear membrane around that collection of chromosomes. So what we're going to do is, in order to replicate our nucleus, we're going to dissolve the nucleus, separate the DNA, and then rebuild two new nucleuses around the existing DNA. Once we start this, those spindle fibers can start to disappear, the nuclear envelope will reappear, the nucleolus is going to reappear, be reformed, and we're going to end up with two distinct daughter nuclei. What you can see here is that we still have the cell membrane intact. Here's the outer cell membrane is still intact. There is still a slight connection between the cytoplasm of the two daughter cells. So the last thing we need to do is undergo cytokinesis. Cytokinesis is the division of the cytoplasm and the organelles. What we're going to see is a slight indent is going to form called the cleavage furrow. This is going to pass around the circumference of the cell. So what happens here is we're actually going to slowly pinch off these two cells. So we're going to separate them, pull them apart, and then start to pinch them off with a ring of actin filaments. So if you remember back to the cytoskeleton, we talked about actin filaments, and we talked about actin and myosin present in our muscle fibers. We talked about actin and I'm sorry, I'm drawing a blank. We talked about actin and myosin. We talked about large filaments, thick filaments, and thin filaments. Actin filaments, those thin filaments, are going to be responsible for forming a contractile ring. Basically, they're just going to form a ring of filaments right around this region, and they're going to pull it tighter and tighter and tighter until eventually we pinch off those two cells. So those actin filaments are going to form a contractile ring. As the ring becomes smaller, the cleavage furrow is going to pinch the cell in half. All right, so that's a very quick overview of mitosis. Please, please, please read through the section and ask me if you have any questions, comments, or concerns. This would be an excellent topic for you to research for those extra credit videos. There are so many videos out there about the processes of mitosis and meiosis. If you check out those videos, you'll be able to see animations of this. You'll be able to see this process in action. So I want to strongly encourage you to check that out. It'll be a great opportunity for some extra credit. So please let me know if you have any questions. You should be able to explain how the chromosome number of the daughter cells compares with the chromosome number of the parent cells following mitosis. I want you to know not only how the chromosome number compares, but tell me about those chromosomes. Are those chromosomes genetically unique, genetically identical? You should also list the phases of mitosis, explain what is happening in each of those phases. You should also be able to describe how the cytoplasm is going to be divided between those daughter cells following mitosis in the process of cytokinesis. All right, please let me know if you have any questions, comments, or concerns.